Hey everybody! Today we're going to be talking about the M-Staphylococcal broth that we're going to be using in our lab. The M-Staphylococcal broth is a selective medium that is generally used to isolate different types of Staphylococcal species. And looking at the media, you might say this looks fairly similar to our basic growth media, and that's because it basically is. So M-Staphylococcal broth is where that we take something like a nutrient broth or a tryptocase soy broth, and we just add 5 to 7.5% sodium chloride in it. The basic thing here is that Staphylococcal species are very resistant to high osmotic factors such as high levels of salt, and most other organisms are not. So normally when we run these tests and we try to isolate the staph, normally from our body or something like that, we tend to just kind of put our samples into these M-Staph broths right away, and in that way we get rid of a lot of things that are not staph. So really when it comes to inoculating the M-Staph broths, we can do it using our basic techniques such as a broth to broth transfer, a plate to broth transfer, or a saline to broth transfer. But most likely what we're going to end up doing in class is that we're going to be using sterile swabs. And in this case here, you'll probably be swabbing things like your nose, the back of your throat, or fomite, and these will then go in separate M-Staph tubes so that we can eventually see what type of staph we have on our body and around our lab. But really, that covers what we need to know about the M-Staphylococcal broth.